Hi there, thanks for your interest in my virtual poster. My name is Pia Rautenstrauch and I'm a PhD candidate in the Computational Regulatory Genomics Lab of Uwe Ohle at the MDC in Berlin. In this poster, I introduce you to the model LIAM that I developed to tackle the complex integration tasks that are posed by multimodal single-cell data. And if you want to learn more about LIAM, you can also consider listening to my talk this Wednesday afternoon in the RegSys COSY. So multimodal single-cell sequencing data really promises to provide unprecedented insights into the molecular state of cells, but at the same time it poses novel data integration challenges. On the one hand, we need to combine data with complementary information that follows distinct statistical distributions, and on the other hand, we need to remove batch effects between multiple samples. And I call these two different tasks vertical and horizontal integration tasks here. While there exist solutions for either of both of these tasks, they're not easily extendable to solve both of these problems at the same time. So to this end, I've set out to develop the model LIAM, which is an acronym for Leveraging Information Across Modalities. LIAM learns a joint low-dimensional data representation of two single-cell modalities measured in the same cell and accounts for batch effects. It currently supports pairwise combinations of gene expression, chromatin accessibility, and cell surface protein measurements. And while I won't have time to introduce you to all the modeling decisions that I took, I want to highlight a few that I consider of particular importance. So firstly, I employ the recently proposed negative multinomial loss to model the chromatin accessibility data. And this loss function was introduced earlier this year by my former colleague Wolfgang Kopp. And concerning batch integration, I make use of the conditional variational autoencoder framework in combination with an adversarial training strategy. And so this is the default mode of LIAM, and it's also depicted in the schematic representation here. So you can appreciate that we combine a conditional decoder, so we feed the batch information to the decoder, and this way we provide a bypass to the model such that there is no need to relearn any batch-related information. And on top of that, we introduce an additional neural network, a batch classifier, that during the training tries to predict the batch from the low-dimensional embedding. And we set up the objective of our model in such a way that the objective of the batch classifier and the objective of the overall model compete, and we make use of a gradient reversal layer to give improvements of the batch classifier back as a negative feedback to the encoder during the training. Let's look at LIAM's performance in practice. I've brought along two use cases. So in this first use case, this is a benchmark dataset from a recent competition where the challenge organizers curated two large-scale datasets, one for site-seq data, the other one for multiome. And one of the tasks was to solve this very task that I just described. And LIAM ranked second for the site-seq data and fourth for the multiome dataset. The results that I'm showing here is a follow-up analysis of mine after the challenge. So we teamed up with the challenge organizers because we found out that one of the batch removal metrics that was used in the challenge, the ASW batch sample, was confounded by the nested batch effect structure of the data. And you can appreciate this by looking at these UMAP representations of the different models colored by different meta-information. Looking at the meta-information sample and site clearly demonstrate there is a stratification by sample and even more pronouncedly by site in the winning submission. And this metric was not able to pick this up because it's based on the average silhouette width and it only considers the closest cluster. So when you have good mixing within the site, you will achieve a high score here, even though you're not able to remove the nested batch effects. There's a simple solution to this problem, which is using either complementary metrics or computing this metric on a different hierarchical level. And doing both of this clearly demonstrates that LIAM and multi-VI outperformed the winner on the nested batch effect removal. I also want to point out that LIAM outperformed its two competitors here on those bioconservation metrics that are dependent on the cell type annotations. And with this, I'd like to introduce you to the second use case, where really this flexible batch removal strategy that we implemented in LIAM can unfold its full potential. So here we have treatment control experiments with replicates available, And what we can do with Liam is to train two different variants of our model using a different variable as the batch variable. So this is what I did here. So the treatment here is in vitro stimulation of T cells. And we train once the model with the replicate, once with the sample. So when we use the sample as the batch variable, we encourage the model to remove all differences between all samples. Whereas when we use the replicate as the variable, we leave a bit more freedom to the model to also pick up subtle biological differences between samples. And this is exactly what you can see in this figure. So when looking at the replicate, we see a very nice mixing of replicates in both models. And when we look at the actual sample ID, we see this strong stratification by treatment and control in the embedding obtained by the model that uses the replicate as the variable. And this is like further exemplified by the cell surface protein expression of our holdout modality. So we use cell surface protein expression as a holdout here. And you can see that secondary effects of the treatment, this is for which CD32 is the marker for, is only retained in those embeddings where we use the replicate as the batch variable, but we see this is like completely blended in and mixed when we use the sample as the batch variable. And this really highlights how Liam can exploit those replicate to disentangle technical from biological variation. And I want to stress the importance of this in light of the dependence of many downstream analysis tasks on this initial low-dimensional embedding. In the future, I plan to extend Liam to more modalities and to the mosaic integration task. And I'm also currently working on a preprint where I further explore evaluation strategies, especially for vertical integration and the comparison of different modeling choices. In a nutshell, Liam is the novel model for the simultaneous horizontal and vertical integration of paired multimodal single-cell data. It outperforms alternative models on a benchmark dataset, and its versatile batch removal strategy really allows it to disentangle technical from biological variation. Yeah, and with this, I want to thank you for your attention, and I'm happy to answer any questions on this platform or via mail or maybe even in person.